Hello, my name is Marco Nievo and I'm the CSO of the Children's Human Foundation Europe. I am awfully sorry, but the first slide of this webinar has no comment at all because there was a fault in the recording. I apologize for this, but right after the first slide, the whole masterclass will be there for you. I apologize again.
And they want to start a family of their own, they are faced with some difficult decisions that they have to make about their reproductive options. Of course, they can choose for spontaneous pregnancy without any genetic testing and to take the risk of having a child affected with NF1 that can be followed up, of course. Other patients don't want to take this risk, and there are several options for these patients. They could choose for a spontaneous pregnancy with prenatal diagnosis by chorionic villus sampling or amniocentesis, and then maybe terminate the pregnancy in case of an affected fetus. But to avoid this difficult decision for termination of pregnancy, there is another option of choosing for in vitro fertilization with preimplantation genetic testing, which we will talk about today. The other options like adoption, foster parenting, use of a gamete donor, or to refrain from child wish are not the first choice for most of our patients. How do patients make a choice? This can depend on several factors, such as, for example, what they know about the condition and complications that could arise in their children, the experience that they have in their self or in their family with the condition, but also ethical considerations and legal regulations play a role because there are differences between countries concerning availability and rules about prenatal and pre-implantation genetic testing. But we believe that it is important for patients to have good genetic counseling about the condition and the options that they have to make an informed choice. I am not aware of any publications about what patients with NF1 think about PGT, but there were some small studies in the past before even PGT was available for patients that looked to the attitudes of patients towards prenatal diagnosis. So a study in 93 stated that 29 subjects who are still considering children, 41% of them wish to have prenatal diagnosis in a future pregnancy, but only 3 of them stated that they would terminate an affected pregnancy. And another study some years later said that 6 of the 17 people planning future pregnancies would want the test with a view to termination if necessary, but others would welcome testing in order to be prepared for the birth of an affected child, but not to terminate. So what is PGT? In fact, it is a genetic diagnosis that is performed on one or multiple cells of an embryo that is created by ICSI fertilization. And it is a technique that can be applied for couples with a high genetic risk for a serious condition. And this condition can be either monogenic with autosomal dominant, recessive, or X-linked inheritance, but it can also be applied for couples where one of the partners carries a chromosomal rearrangement like a reciprocal or Robertsonian translocation. One of the premises for this to be able to do this is that the mutation has to be known. So it's not enough to have the clinical confirmation. And the purpose of the technique is to transfer only unaffected embryos so that the couple knows already at the moment that they have a positive pregnancy test that their child will not be affected with NF1. There are different techniques to do this, and in the past, usually the biopsy was taken from day 3 embryos. So in the cleavage stage, 1 cell of the embryo was removed with a biopsy and was examined for the NF1 mutation. Nowadays, the golden standard is to do the biopsy somewhat later on the 5th or 6th day, and we remove about 5 or 6 cells of the trophectoderm, and these cells will be evaluated. So a little history on PGT. It was already by the end of the 80s that it was tested. So it was reported in 1989 that it is possible to do a genetic diagnosis on a single cell that is removed from a day 3 cleavage stage embryo. And in 1990, the first report was published about pregnancy that followed a genetic diagnosis on a blastomere cell. This is a reference of a publication in 2005 from our colleagues in Brussels, who have been very active also in the PGT field, and they described their experience with PGT for NF1. So at that time, the technique that was used was multiplex PCR. So before the treatment, the IVF treatment can start, there is a genetic preparation necessary, and we will need blood samples of the couple and also from an affected reference index. And this can be a parent or an affected child. So in this example, we have the male partner that is affected with NF1, and he inherited the mutation from his mother. So using microsatellite markers that are linked to the NF1 mutation, we can do multiplex PCR to detect in the embryo the microsatellite markers that are linked to the NF1 mutation. This is a review about the evolutions that have taken place in this field, and the first PGT, they were locus specific, so they only looked at NF1 locus, but we didn't know anything else about the genetic content of the embryo. Nowadays, most of the labs have moved to genome-wide techniques with haplotyping to analyze the genetic content of the embryos. So in our lab, we use PGT haplotyping via SNP arrays. So we also need a PGT preparation step. So you see on the right, the pedigree. Here it is a couple that has already an affected son, and we can deduct that the affected son inherited the yellow haplotype from his affected mother. So if we see this yellow haplotype back in the embryos, we know that the embryo is affected. If we see the other, the green haplotype, we know this is the unaffected haplotype, and this embryo will not be affected. So an embryo cannot be transferred in case that we detect the affected haplotype. So we know that the embryo carries the NF1 mutations. But since this is a genome-wide technique, we can also detect aneuploidies like trisomies or large segmental chromosomal abnormalities. And we will also select against these abnormalities because they will not implant, these embryos will not implant, or they will be born with congenital malformations. Sometimes it happens that it is not possible because of technical reasons to have a result for the embryo, and then the embryo will also not be transferred. This is a very complex procedure, and theoretically there is a small risk for misdiagnosis, which is estimated to be less than 3%. So as we know, a lot of our patients have their NF1 due to a de novo mutation, so we cannot directly use DNA from a reference index. So we have to do an additional workup for which we use long-range PCR. So we create a long-range PCR around the NF1 mutation, and you can see the mutation in red, which is linked to the SNPs that we can see back in the father. So in this case, we can say that the mutation has occurred on the paternal allele, which is in light blue in this example. And so afterwards we can do the haplotyping as for other cases, and we will select against embryos that inherit the haplotype that is in light blue. So once all preparations are done, the couple can start with their IVF treatment. So the female in the couple will receive a hormonal stimulation by the fertility doctor, and then she will have to come for a pickup of the oocytes. The sperm has to be processed, and the fertilization will be done by ICSI procedure. We can follow the development of the embryos in the lab. And as I told you, we used to do the biopsy on the blastomeres of a day 3 embryo, but nowadays we always do trophectoderm biopsies from the day 5 embryo. So the genetic analysis with the SNP array can be performed on these biopsies, and in the meantime, the embryos are frozen. We will know the result of the haplotyping by the next menstrual cycle, and then we can make the distinction between the embryos that are unaffected and those that are affected. And an unaffected embryo can be thawed and can be transferred to the womb. So as I told you, it is a genome-wide technique, and if there is any large chromosomal abnormality, we will also detect it, and we will also select against this. So embryos with aneuploidies will not be transferred. And then we zoom in on the NF1 locus. In this example, we have a family where the mother is affected with NF1 and they have already an affected child. And with the pre-PGT preparation, we could deduct that the child inherited the haplotype in red from his mother. So this is the affected haplotype. So you see two plots from embryos on the above on the left. You see the plot, and the NF1 locus is where the yellow dotted line is. So for this embryo, we see the red haplotype. We know that this embryo inherited the mutation. For the embryo on the right, we see that the embryo inherited the pink haplotype, and this embryo will be healthy for the NF1 locus. One of the difficulties with PGT is that it doesn't always succeed because there are different steps in this procedure. So as a mean, after the hormonal stimulation, we can have about 10 oocytes after pickup, but this will not all be mature oocytes. The fertilization will not work for all these oocytes. These embryos will not all grow and develop until the stage that we can do a biopsy. And then obviously we have to select against those that inherited the mutation or have an aneuploidy. So this means that as a mean, we have about 1 to 2 embryos per cycle that are transferable. And then we also have to be aware that not every transfer of an embryo will result in implantation and an ongoing pregnancy. You have the plots of the chances for a positive pregnancy test and evolving pregnancy, which were some years ago about 25 to 30%. But nowadays, during the last 2 or 3 years, we have had some changes, and now we have about 50% positive pregnancy tests after embryo transfer, and this will result in about 40% live birth rate per embryo transfer. This is the top 20 of indications for PGT in Leuven, and you can see that NF1 is in the top 5 as an indication for PGT. And on the right, you can see that the autosomal dominant conditions are the most frequent conditions where we do PGT. So we looked into our data with PGT for NF1 in Leuven, and we have had 39 couples that came with a question to do a PGT preparation. In 17 of these couples, the male was the affected partner. In 22, the female was the affected partner. 24 of the 39 couples were familial cases, and 15 cases were de novo. Since the de novo workup is more complex, it doesn't always succeed, but still in 13 of the 15 de novo cases, we were able to offer PGT. And then if we look on the right to the PGT cycles, we see that 24 of these couples really started a cycle. In total, we had 45 PGT cycles for these 24 couples, and we had 210 embryos that were biopsied and that were examined. So this is an average of 4.6 embryos per cycle and an average of 2 cycles per couple. On this slide, you can see the embryo results plotted as a percentage. So about 43% of the embryos were affected, so carried the affected haplotype. About 36% were unaffected, and you see that it is an important percentage. It's about 16% of embryos that are labeled as abnormal. This means that these embryos carry an aneuploidy, so this is not distinct from the NF1 mutation, but we detected some abnormality for which we decided not to transfer this embryo. And then a very small proportion of embryos, they have inconclusive or no result. From these embryos, we had until now 40 embryo transfers carried out in 14 patients, which resulted in 12 live births in 10 patients. So 2 patients already had a second child after PGT. This slide is to show you what is the most important prognostic factor, because most of these couples, they don't have a fertility problem, but it seems that it's only the maternal age that is confirmed in different studies that will really have a prognostic factor on the success chances. So you see after the age of 35 and certainly after the age of 40, that the chances that this procedure will succeed will be a lot lower. Another pitfall is that patients tend to think, well, our embryo has been checked and everything is okay and nothing can happen. We will have a perfectly healthy child. Of course, we have to be aware that there is a very small risk of a misdiagnosis of the PGT. And then we also know that every healthy couple has a risk of about 3% to have a child with a congenital condition or malformation. And this risk remains, except for the aneuploidies that we can detect with our technology. patients are often concerned about risks for their child after this procedure, because it is an invasive procedure. We take a biopsy from a growing embryo, and there have been numerous studies that showed that there is not an increased risk for a congenital malformation in children born after PGT, and also follow-up studies in young children did not show any differences in their cognitive or psychomotor development. So it seems to be a safe procedure. However, we are always searching for better techniques. So I already told you the shift that we have seen in the time of taking the biopsy, where it used to be on the 3rd day. We now do the biopsy on the trophectoderm in the blastocyst, but that there are studies for what we call non-invasive PGT, so to try to amplify DNA from the blastocyst fluid in the blastocyst, or even the IVF medium, to do the diagnosis on this material and to not have to biopsy the embryo. So in conclusion, PGT is a good method for patients with NF1 to avoid transmitting the condition to their children without a need for prenatal diagnosis and termination of pregnancy. But it is a very complex procedure with limited success rates, which are, however, improving, and a small risk for misdiagnosis. So it's crucial that patients have good counseling before they decide to go for this treatment. So if there are any questions. I don't hear you.
You're still muted, Marco.
I should unmute myself. That's a good idea. Thank you very much, Ellen, for this very, very clear presentation. We are starting to have questions coming up in the chat. One is from Lars Eriksson, whom I assume is not a medical professional, given the tone of the question. It's slightly unrelated, but still interesting. Basically, it's a very long question. I'm going to make it shorter. This person had preimplantation genetics done on in 2010. And apparently one embryo turned up to be trisomic for trisomy 21. And his question is, and he talks about his own age and the age of his wife. And he asks if there's an increased risk of somatic variation as the age goes, as the age of the parents increases.
Yes, this is what we know, that the risk for trisomy 21 or other trisomies increases with the age of the female partner, and this is also what we can detect in our embryos. But even in young couples, sometimes we can find an embryo with a trisomy, but the risk is definitely increasing with the age, and this is.
And this risk is mostly correlated to the age of the mother, not that of the father.
Yes, this is related to the age of the mother. Yeah.
So I think Lars's question is answered. Another question from Mark Sterkel, who asks, and actually I was interested to know that as well. He asked, are the genetics tests available in all countries? And my question would be, do you know of any country where these tests are not available at all?
Yeah, I think I don't know all the regulations in all the countries, but I think there are countries where this is not available, but I cannot say from the top of my head where it is available.
It's a very complex technology, and you need to have an advanced medical system to bring this to the patient. That's number one. And number two is that sometimes in fertility clinics this possibility is offered, but it is not reimbursed by the local health insurance system. So there are still a number of barriers for patients who want to use this technology, because it's not available in every country and it's not reimbursed in every country. In Belgium, we are lucky that 6 cycles of IVF with PGT are reimbursed by our local health insurance system. But 6 is a lot, and it's more than is usually reimbursed in other countries, where sometimes only 3 cycles are reimbursed. And if you are not pregnant after 3 cycles, then you have to pay for it yourself. And it's a very expensive technology if you have to pay for it completely by yourself.
Thank you. A question for Rebecca Brown. For the de novo NF1 patients with a clinical diagnosis of NF1, but negative genetic testing, what is the best path forward to identify a mutation?
Yes, in this case, if we do not find a mutation with the regular sequencing, we go to analysis at the level of messenger RNA. So we don't look at the DNA, but we look at the RNA. And there we have a chance of a couple of percent that we find a specific splice mutation that is not clear when you look only at the DNA level. And then if that is also negative in the blood, we have to consider mosaicism. And if there are if there is some clinical evidence for mosaicism, meaning that some of the café au lait spots or neurofibroma cluster in one region of the body and are not present, let's say, dispersed all over the body, then we might have to analyze a café au lait spots or neurofibromas, because there we will certainly find the responsible NF1 mutation. Of course, when you don't find the mutation in the NF1 gene in a person with multiple café au lait spots, there are alternative diagnoses, and you should also analyze the SPRED1 gene and a couple of other genes. So at a certain moment, you have to also assume that potential other conditions that are associated with café au lait spots have to be considered in the diagnosis. But usually nowadays in many labs, they use gene panels where they screen for most conditions that are associated with multiple café au lait spots. But then, of course, if you don't find it, it is the best thing to do is to contact a center of expertise where they have expertise in DNA analysis or molecular diagnosis for NF1.
Thank you. I don't see any more questions from the audience, so I'm going to come up with one myself, because this issue of genetic, pre-implantation genetic testing, I suppose, is a very delicate issue to approach with the patients. And I was wondering what your, what advice you could give to anyone in the audience when it comes to actually suggesting to people and offering the genetic testing, this kind of procedure to people. If you were being very active, or you were just wait for them to come and ask.
Yeah, my advice would be in young patients, young adults, just to give them the information that they have to come for a consultation at the moment that they are considering to have children, and that we'll talk about the options at that time, and we will go through all the options. And of course, it is up to the patient to make his or her own decision together with a partner. But yeah, usually when we see young adults, we already touch this subject, just that they know at the time they are considering.
Oh yeah, you proactively do it and systematically with all your patients.
That's the recommendation, to systematically touch this issue without going really into detail in every different option, but to say that there are options and that they should come to discuss these options if they want to. so that everybody is aware that there are options, and if they are willing, they could discuss these options. It doesn't mean that they should use one of those options, but they should be aware, and we recommend to systematically touch this issue and mention it in young adults who potentially want to get children in the future, or in adults that we see for the first time who are of childbearing age.
Sounds like a very good policy to me. I was wondering as well if the couples that approach you to actually do that, do they, I mean, is it mostly very severely affected cases, or it's any kind?
Yeah, I think this is also part of the counseling, because it's, we know that the phenotype in the child can be different from the phenotype in the parents. So we have also parents that have a very mild phenotype that come with this question because they are aware that the phenotype could be worse in their children. So it's not only the patients that have a severe phenotype themselves that come with this question. No.
And sorry, I may have missed that during the talk, but do you screen for all genes when you do it for NF1, or do the parents have to ask, you know, specifically for other genes to be screened for it as well?
So in the embryos, we look at the chromosomal abnormalities, and we look at the NF1 locus, but we will not test for other monogenic conditions in the embryos, but we have some routine genetic tests that we do also during the pre-PGT preparation phase. So we test all our couples or one of the partners for cystic fibrosis, and also for Fragile X and SMA. So these are reimbursed in Belgium for all couples entering an IVF program, and so also a PGT program. So we could detect that a couple has another condition, but we don't test a lot of conditions. However, there is also the possibility of an extended genetic carrier screening, which tests for carriership of more than 1,000 autosomal recessive disorders and 100 X-linked disorders, but this is not reimbursed. So this is really optional. Patients are informed that they could do this test also, but this is during the pre-PGT phase. So it is on the blood of the parents that we test this, and if they choose for it and nothing comes out, we don't test anything else in the embryos.
But in the embryo, Marco, we systematically look and test all SNPs and all chromosomes, because that's part of the technology that we use. So you cannot opt for, let's say, only the NF1 gene, because it's part of the whole analysis package in the technology that we use. And by looking at other chromosomes where we could have a chromosomal abnormality, not a gene error, but a chromosomal abnormality, so it's like a trisomy or a monosomy, we can select for the best embryos that have the highest growth potential, because it is of no use to transfer an embryo that has 3 or 4 chromosomal abnormalities, but where we know that the NF1 gene is not mutated, because we know that this embryo will have very, very few chances to grow into a pregnancy, and then we don't use this embryo. But this is standard practice. This is discussed up front with the couple, that we see more, that we also see the chromosomal abnormalities, and not only the NF1 gene inheritance pattern, but also the others. And they all agree that we, of course, do not transfer embryos with a chromosomal abnormality that we see, let's say, just by coincidence. But we know that even in young couples, as Ellen said, it is not infrequent, so it is frequent to see embryos with a chromosomal abnormality. And that's the reason why not every embryo that is transferred will grow into a pregnancy. There are, of course, other reasons that will have an influence on this, but this is the reason, or this is one thing that we can eliminate by looking at them. Of course, we can only say something about the cells that have been analyzed. And if there is mosaicism in the embryo for a certain abnormality that is not present in the cells that are part of the biopsy, we will not know this.
Yeah, of course.
Yeah.
Of course. Yes. I was wondering also if you could dive into more detail about the techniques. You were talking about alternative techniques to you know, fully fledged embryo biopsy. I wondered if you could, you know, explain to us at what level of, you know, how advanced these are. Are they still so experimental that that you'd rather not use them for?
No, for diagnostic reasons, we're not ready yet to use these new techniques. But however, there is interest in the field and studies are being done. But I think during the first years, probably we will stick to the trophectoderm biopsy.
Yeah, I think for the moment this is the safest, Marco, because we know that when an embryo grows, that not every cell continues to grow, and some cells die. They liberate their DNA in the culture medium, and you could look at that DNA to see if there is something with the НФ1 gene or with another gene that you want to analyze in that particular embryo. But because these are cells that are dying, we don't know how normal their chromosomes are, and if it's going to be reliable to do an analysis on this free-floating DNA from dying cells. And so this has to be resolved, this issue, and we have to know that these cells have a good DNA composition and are representative of the whole embryo before we could even start thinking of using this technology. It is not a difficult technology to use free-floating DNA, because we do this all the time for free-floating DNA in the maternal plasma of pregnant women to do the non-invasive prenatal testing. And of course, it's just a small adaptation of the technology to do this for pre-implantation genetic testing. But we have to know that this is reliable and that this DNA that can be found there is sufficient to do an analysis and is representative of the whole embryo.
Yeah. Yeah, it's also because I mean, technically there wouldn't be any need to develop more technologies. I mean, the embryo biopsy seems to be very well validated and very safe. So is this mostly for ethical reasons that why are we looking for these alternative ways?
It's because it's still conceived as an invasive procedure, and sometimes we have embryos that don't survive, and we don't know, is it because of the biopsy or is it because of the embryo itself. So yeah, people are always looking to possibilities to do diagnoses that are less invasive, but
and destructive. I mean, this is
very well for the moment.
Yeah. We have a question from Ana Elisabetta Pires. Do you analyze all the 5 and 6 cells independently or as a whole?
As a whole, yeah, we analyze them as a whole. So we don't analyze them independently.
And also she asks, and I wanted to ask that too, but I'm happy somebody else does it. Can you give an idea about the cost of the procedure in Belgium, if somebody had to pay of their own pocket for it?
Yeah, I know that I'm not that good in numbers, but I think if for the couples that have the reimbursement, it's about 800 euros that they have to pay for themselves. If they don't have any reimbursement, it's, I think for the total, I think the PGT is about 2,000 euros, and then the PGT per cycle, I think about 3,000 or something.
1,600.
1,600.
So for the genetic testing, the preparation is 2,000 euros, and for the genetic testing on the embryos, independent on the number of embryos that have to be tested, is 1,600 euros. But on top of that, if IVF is not reimbursed and you have also to pay for the IVF cycle, you have to pay an additional 2,000 or 3,000 euros on top of that for the IVF cycle, to pay for the hormones that are used in the stimulation, and to pay for the IVF media and the culturing of the embryo during these days when they grow, and also for the freezing and thawing of the embryos. So if nothing is reimbursed, it becomes a quite expensive technology.
Very expensive. It's very, very expensive. There's actually a statement in the, not a real question, but a statement from Megan Wise in the chat, which says that it seems that, you know, a genetic disorder of 1 in 3,000 would warrant testing, prenatal testing anyway, regardless of the, you know, would warrant testing, prenatal testing on the children, which is it's a bit unrelated to that, but it's to the topic of the conversation today. But Megan made this statement, and I wanted to make sure it was put on the table.
Yeah. So if you don't have any clue, if both parents do not have NF1, then of course it doesn't make sense to screen a pregnancy for NF1, because the chances you will find an NF1 mutation in such a pregnancy is 1 in 6,000, because that's the de novo mutation rate that you find in couples who themselves don't have NF1. But in these cases, of course, one of the partners has NF1, and their chance, of course, for the child is 1 in 2, is 50%, and not 1 in 3,000, of course. And that's a completely different situation.
Yes. Actually, have you ever had couples where both were NF1 affected by NF1?
Excuse me?
But have you ever had couples where both parents were affected?
No.
Not that I remember, no.
No.
I don't think so.
Not in Belgium.
Yes. Well, I don't see any more questions. Is there anything other that you'd like to highlight around, you know, around your experience with pre-implantation genetics in your clinic?
Well, I think if it's available in a country, then I think it's worthwhile to try to convince the authorities to reimburse it, because of course it's an expensive procedure, but the alternative to care for a child with NF1 that might have a number of problems is also expensive. So I think that at the level of the health insurance, it's better to reimburse pre-implantation genetic testing from a health economic perspective. So I think this could be used as an argument in countries where this is not reimbursed because of financial reasons. If it's not reimbursed because of ethical reasons, then it becomes, of course, a lot more difficult to argue.
Then we have another question, which is slightly unrelated, but still, if a normal mutation rate is 1 in 6,000, how come is the prevalence 1 in 3,000?
Because half of them inherit the mutation from their parents, and that's 1 in 6,000. And the other half have a de novo mutation, that's also 1 in 6,000. So you end up with 2 in 6,000, which is 1 in 3,000. That's the math behind this.
And another question, which is slightly unrelated. I mean, but still related, not directly related to the topic, is from Anna Elisabetta Pires. Can you interrupt a pregnancy in Belgium due to an NF1 mutation?
Yes, it's possible. So it's possible. So it's legal. You can do this. But as Ellen already showed in one of the slides, even parents who decide upfront that they would go for a pregnancy and then have a test during pregnancy with pregnancy interruption, they quite frequently also change their mind during the pregnancy when it turns out that the fetus is affected, even though they had decided before the pregnancy they would terminate. Many of these individuals would still choose to keep the pregnancy. That's our experience. So we have very, very few requests for pregnancy interruption.
Stephen Sherd, who congratulates you for your presentation.
Thank you.
That's all. It's a statement. He says this is a frequent topic of conversation with older adolescents and young adults with NF1 he works with. There's a question from Sandra Tolle. How do you estimate the risk for NF1 women with a mosaic NF1 to have an affected child?
Over there. Can you repeat the question?
Yes, the question is, how do you estimate the risk for an NF1 woman with a mosaic NF1 to have an affected child?
Yes, we don't know. There are no good data available. We don't know if the, let's say, the proportion of the body that is affected or the region of the body that is affected has any influence on the actual transmission rate. So we have very, very few data on this aspect. The only thing I can say is that in our series, we have indeed a couple of parents who were mosaic and who had an affected child. So it is not a risk that is below 1% or so. It's a substantial risk, and I estimate it somewhere between 5 and 20%. But that's as far as we can go at the moment. And we really do need more data on this. And we need, I think, with different centers who have data on mosaicism and on adults with mosaicism and on their children to see what the actual transmission rate is. The only way of estimating this is in males, and we quite often, when we have a male individual with a mosaic NF1, test the sperm cells, and there of course we can see if it's present in 1% of the sperm cells, then the risk is 1%. If it's present in 25% of the sperm cells, the risk is 25%. If we cannot find it with the actual techniques, then it means it is maximum present in less than 1%, then the risk is very, very low. And then
but you do recommend to all your patients, regardless of whether they're mosaic or not, to undergo, I mean, you do offer all NF1 patients the possibility to do genetic pre-implantation testing.
So pre-implantation testing in mosaic individuals is complex because you don't know what the transmission rate is going to be. And if it's a very small risk, so in males you can look at it in the sperm, but in females it's of course not possible to have an idea how many of these oocytes will have the mutation. And then sometimes it is offered when several tissues are affected, such as blood and neurofibromas and the melanocytes in the café au lait spots. But it makes it somewhat more difficult to find out which of the two alleles the mutation occurred. So it takes a longer preparation time. to figure this out. And then you know, of course, that you will block all embryos with a specific haplotype from transferring, but you know that maybe most of them don't have the NF1 mutation, because you're not looking at the mutation itself in the embryo, you're looking at the haplotype that is associated with the mutation. But in mosaics, we know that the same haplotype is sometimes associated and sometimes not associated with the mutation. So it makes it a little bit more complex. But it is not impossible.
Another question from Lars Eriksson, who asked a very basic genetics question, is if my daughter has NF1 and her partner does not, what are the chances of producing embryos without NF1?
It's 50%.
Yeah, it's 50%.
Yeah.
It's one of two.
Yeah.
I don't see any other questions coming up. And we are almost at the hour. So I want to thank Ellen and Eric for this very clear class and for answering very thoroughly all the questions today. And thank you, the audience, for participating and for being here very numerous today. There were 30 of you online. I have a question in the chat asking when the recording will be available. The recording will be available in English in the next few days on the YouTube channel. I will email all participants when that is done, and that will be followed by versions in all the other languages, which are French, Italian, German, Portuguese, Spanish, and Russian. And unless there aren't any more questions, I thank everyone and hope to see everyone again, for our next masterclass.
Okay.
Thank you.
Thank you.
Bye, everyone.
Bye.
Bye.
Bye, Marco.
Bye.
Bye. Thank you very much. Bye.